Hi, my name is Pablo Vergara. I am a postdoc in Sakaguchi-lab in the International Institute for Integrative Sleep Medicine. And today I am going to share with you my last finding on how the adult-born neurons operate during memory processing. Most brain regions in mammal cannot produce new neurons during adulthood. One exception to this is the hippocampal dentate gyrus, in which new neurons are produced throughout life. Interestingly, several optogenetics and ablation experiments have shown that these adult-born neurons are necessary for the processing of memories. My project is to identify the exact mechanism by which these adult-born neurons support memory processing. One simple mechanism could be that the adult-born neurons operate as engram cells, which is by directly holding memories. If this is the case, the activity dynamic of adult-born neurons should support at least two properties. One, a specific population should be activated during learning, and two, the same population should be reactivated when animals remember the encoded memory. Neurons displaying these engram properties have been identified in several brain regions, such as the amygdala or the prefrontal cortex, but most importantly, also in developmentally born granule cells. This naturally raises the question of whether their adult-born counterparts are also displaying these engram properties. To address this, we imaged the calcium activity of individual adult-born neurons in freely behaving mice during the learning of a contextual fear memory during subsequent memory consolidation and retrieval. For example, here I show the calcium activity of 6 adult-born neurons in the home cage before conditioning and in a new context before and after receiving an electrical shock. So first I wanted to see what is happening during the learning period. This heat map shows the activity of individual adult-born neurons. Each row is a different adult-born neuron. As you see here, when animals are exploring a new environment, some adult-born neurons increase their calcium activities. You can also see that this increase in activity is reverted after the shock experience, indicating that fear learning predominantly leads to a decrease in adult-born neurons' activities. So are these adult-born neurons that are active during learning being reactivated during memory retrieval? To answer this, here I show the average activities of all the recorded adult-born neurons in the session corresponding to the home cage learning and retrieval. You may notice that the population activity in the home cage and during memory encoding are very similar. This means that predominantly active neurons in each of these sessions are virtually the same. Surprisingly, and in contrast to the last result, the population activities before the consolidation period were fundamentally orthogonal to the activities during memory retrieval. This result means that, while a similar population of adult-born neurons is engaged during learning, a fundamentally different population emerged during the memory retrieval period. To investigate what is happening during the memory consolidation period, we divided the population activities into 15 minutes bin. Then, we calculated a remapping index that measures the degree of similarity of each time bin to the first and the last time bin of the memory consolidation session. This remapping index ranged from -1 to 1, taking a value of 1 when population activities perfectly match the first bin, and a value of -1 when it perfectly match the last bin. As you see here, remapping indices were strongly correlated with time, indicated that adult-born neurons' activity gradually remapped during the consolidation periods, rather than abruptly changing during memory retrieval. This remapping was detected regardless of whether mice were awake or asleep. To investigate if this remapping process represents a global phenomenon or affects a discrete group of adult-born neurons, we identified the adult-born neurons with activity significantly correlated with time. This revealed two subgroups of neurons, one decreasing their activities and another one increasing their activities. The neurons with significant correlation represent roughly 25% of the total adult-born neurons. Notably, the adult-born neurons decreasing their activity were predominantly active during learning, while the adult-born neurons increasing their activities were predominantly active during the memory retrieval. This suggests that activity remapping predominantly involves the cells that are active during memory encoding and retrieval, and therefore the adult-born neurons that will be relevant for memory. In summary, we have found that the population of adult-born neurons active during memory encoding does not overlap with the adult-born neurons active during memory retrieval, and that this orthogonalization of adult-born neurons' activity is caused by a remapping process occurring during the memory consolidation period. I am Pablo Vergara. Thank you very much for listening to my presentation. A special thanks to the people shown in this picture and the funding agencies that support this project. Please contact me if you have any question or comment.